Hello, today we are going to discuss how to use Punnett squares. So starting, what is a Punnett square? So a Punnett square is the diagram in the top left. It is a square diagram that is composed of a grid of usually 4 boxes and is used to calculate and depict all the combinations and frequencies of the different genotypes and phenotypes among the offspring of the cross and birds with Mendelian inheritance. So long, complicated definition for just saying it's a grid usually of 4 boxes. What we put in those 4 boxes are genotypes. So that is the genetic constitution of an individual organism. So when we have genes from the father to the mother, and then their father's genes and their mother's genes, basically we use a letter of some kind to, it's kind of like a variable in math to basically represent what their gene is. And then the phenotype is just not what is genetically happening inside them, but what is physically going to be seen. So, as that definition says, it's a set of observable characteristics of the individual resulting from the interaction of its genotype with the environment. Okay, and here is basically how you can kind of distinguish between genotype and phenotype. So, for example, if you were talking about eye color, okay, and the father's genotypes that genotype that he has is he has a capital B for a brown allele, and he has a lowercase b for a blue allele. So that father from his father and his mother, when their sperm and egg came together, he ended up getting one brown eye allele and one blue eye allele. So that's why the father has both a brown eye allele and a blue eye allele, a capital letter B and a lowercase letter b. The mother also happens to have one brown eye allele, a capital B, and one lowercase b for a blue eye. Okay? Now, when they mate, their child is either going to end up having one of those 4 genotypes in the middle. They are either going to have both brown eye alleles, they're either going to get a brown allele and a blue eye allele, or they're going to get both blue eye alleles. Okay. And you don't know that, that's why we use the Punnett square to figure out kind of what is the likelihood that each of those will happen. Now, that is what the genotype is, which is a little more complicated than the phenotype, which is just what you're physically going to see. So, if brown is dominant to blue, if there is any brown allele present, that means the eyes that are going to be expressed physically are brown. So we can see here that 3 of those 4 boxes have a brown eye allele. So 3 of those 4 boxes are going to make brown eyes. The only box that's going to make blue eyes is the lowercase b, because there is no brown eye allele to basically dominate it. Now, for the record, I should say this, eye color is a gene that is actually more complicated than the simple dominance, but some features of eye color are like the dominance that is being presented as Mendelian genetics. For now, it's just a good example that it's easy to visualize and comprehend. Okay, so Punnett squares. When meiosis generates sex cells, those sex cells each have one allele of a pair. When those sex cells are fertilized and unite, the allele from the father and the mother come together to be the alleles of the offspring. Now, in this situation, the father received an allele for brown eyes from his father and an allele for blue eyes from his mother. The mother received an allele for brown eyes from her mother and an allele for blue eyes from her father. This means that each of the parents have a brown eye allele and a blue eye allele. The parents each have brown eyes because the brown eye allele is dominant to the recessive blue eye allele. For meiosis, the sex cells in this instance will either carry a brown eye allele or a blue eye allele. It is completely random as to which alleles will unite together during fertilization. The Punnett square displays all the possibilities as to which the alleles can unite. And we can see we have also calculated calculated genotype and phenotype ratios. So, for example, there's one box with 2 uppercase B's for brown eye alleles, 2 boxes with a uppercase and a lowercase b, and 1 box with a lowercase b. That calculates to a 25% likelihood of getting 2 uppercase B's, 50% of getting 2 uppercase B and a lowercase b, and 25% chance of getting both lowercase b alleles. Now for the phenotype ratio, you get 3 out of 4 boxes will provide you with brown eyes, while 1 out of 4 boxes gives you blue eyes, which is the same thing as saying 75% chance of getting brown eyes, while there's a 25% chance of getting blue eyes. So here I have a list of rules as to how to get every Punnett square problem you are ever given, how you can solve it and get it right every single time. So first you draw a Punnett square, and then you decide on a letter to use to represent the genotype. So no matter where you're given, eyes, hair, skin, whatever it is, it does not matter what letter you use to represent the genotype. However, it should be related to what gene or allele you're representing in the Punnett square. So for when we were talking about eye color, I chose to use B's because the eye colors are both brown and blue, which each start with B. But I could have also used, for example, E's because we are representing eyes, or C's because we are representing color. The capital letter is always the dominant allele, and the lowercase letter is always the recessive allele. A capital letter is always written first. So then you're going to place the alleles of the mother on one side of the Punnett square and the alleles of the father on the other side. You're going to cross them through the Punnett square just like you would a battleship, doing the top column and then the horizontal row, have them come together. A lot like the codon charts too, actually. And then now you have all the genotype and phenotype possibilities. Carrying on this, so when Mendel was studying pea plants in their breeding process, he noticed similar offspring being generated when certain kinds of plants were bred. The main generational nicknames known are parental or P generation, first filial or F1 generation, second filial or F2 generation. Now, on the right are a lot of the different traits that Mendel studied. And the first one we're going to look at is the stem height. So basically, a tall plant is dominant to dwarf or short plants. Here's some more specific definitions for you to read, but the two I'm going to talk about with you are heterozygous and homozygous. So homozygous is just describing when the genotype, basically, they are both dominant alleles, so they are either both capital letters or they are both lowercase letters. Okay, that is homozygous. Homo means same, so they are the same, either dominant allele or the same recessive. Heterozygous, hetero means different. Heterozygous means that one allele is going to be dominant while the other allele is recessive. Okay, so homozygous, both alleles are identical. Heterozygous, each of the alleles are different. Okay, so the father's genes and the mother's genes. So with the parental generation, you are always crossing a homozygous dominant with a homozygous recessive trait. So we're crossing, let's say, a male plant that has both 2 uppercase T's representing tall, and, for example, the mother has 2 lowercase t's representing short. When you cross those through the Punnett square, you get all of the offspring are going to get 1 tall allele and 1 short allele. And that generation is known as F1. So F1 is just the results of the parental generation. That's all F1 is. Now, if you take an F1 plant and you cross it with another F1 plant, so a big T, little t, and another capital T, lowercase t, which we can see there with the father and the mother. When you cross those through a Punnett square, you get one plant that will be homozygous dominant for height, another plant that will be homozygous recessive for height, and then two plants that are heterozygous. They have one large T allele and one short T allele. And you can see that in the image below, that being crossed as well. And then lastly, there is the F2 generation, which is literally just the results of the offspring of the F1 generation. You can see that there as well. On the right side, another example was that yellow peas were actually dominant to green peas in the pea plants that Mendel was studying. So you can see just another example of the pea F1 and F2 there. Thank you for watching. Please share and subscribe, and remember to spread